Come on.
Okay, good morning. Welcome to UC Riverside, everybody. This is a very nice, bright day, and this would have been a great day to be on campus and have an actual symposium. But instead, you get to enjoy the comforts of your own home office, wherever that may be. But we are very excited to have everybody back. Um, really can't believe it's been 3 weeks since I last sat in front of my computer in front of all of you. Except now this is going to be a little different because this is a webinar, and now this time you guys will be the presenters. So we're all very excited, and just hopefully you guys had a really good experience over the past 3 weeks, attempting to work in groups and communicating virtually, as well as getting a taste of what research was about. And so this project started about 8 years ago, and we started off with only 24, maybe 23 students from just one community college. And it was an opportunity to try to give this particular experience to community college students. And as you know, I was a community college transfer too, and opportunities like this to participate in research simply didn't exist. I did not think I would ever work with another university while I'm at community college. I believe most of us think, hey, we have to take our classes and do well, and the real work doesn't begin until you transfer, and that's so far from the case. And I believe you could do so much more at community college, and this is exactly why we've grown this program to where we are. And so this is the 9th annual Sequence to Success DNA Barcoding Challenge. It's hosted by many, many different people here. However, most importantly, it's that all of you as students have taken the time to dedicate your 3 weeks. And I know you guys are all struggling, as well as it's a lot of challenge to balance your classes and any other extracurriculars, work, perhaps family, to work on this project. And I want you guys to all know that we genuinely and deeply appreciate your dedication. So today's presentations is a culmination of all the work that you've done. We've actually accepted, or we've gone through everyone's abstract, and Gina, she's going to be our Wizard of Oz in the back end here, uploading all of your presentations, and she's the one who also created the abstract booklet that we've actually shared. So I wanted to go over a few very brief details for you guys in terms of presentation. So for all the presenters, we will be promoting you to the panelists. As of now, you guys are all attendees. And once you're in the panelists end, you'll be able to simply speak, and we'll go through our progression for the presentation. If you need to go to a next slide, all you do is say, next slide, please. Or if you come to a pause, Gina is going to be smart enough to click to the next one. But if she happens to go beyond what you had hoped, you can just request for to go back one slide. For everyone in attendance, this chat function is entirely open. The expectation is all students or all groups will have 10 minutes to present. Of those 10 minutes, 8 minutes will be their actual presentations, and 2 minutes will be reserved for questions. If time goes over a little bit, that's perfectly fine. We're not under any strict timeline. We do have some buffer scheduled into our program. But yeah, when it comes to asking questions, you can type into the chat, or you can raise your virtual hand onto the on your features here. If you raise your virtual hand, you can actually answer live. We'll just have to unmute you. And secondly, there is a Q&A function that's part of the webinar format here. You could try to type some of your questions into there, and we'll hopefully have the group members address it live if you happen to have questions during their presentations. Okay, so without further ado, I also have one of our instructors and the original, one of our originally founding members of this entire program, Dr. Jim Burnett. He's dedicated a lot of his time, and I still remember the first day we just sat in a room trying to figure out what type of project can we offer to community college students, and something that's fulfilling. It provides a lot of scientific introduction into scientific research, and this is just dipping your toe into what it's all about, and also understanding what is research and how does it affect you? What is the significance? And what are some real world applications? So that's one thing that we changed up a little bit this year, is in addition to having you run a project from A to Z, of starting off with a sample and running a lot of experiments to get data, analyzing that data and coming to a conclusion, we also asked everyone to think a little bit outside the box and to explore what possibilities there can be with the techniques that you've used. So we're really excited to hear everybody's discoveries. But in any case, Dr. Burnett will have a few words to say to everybody as well. So Jim, if you would like, you can turn on your camera and take it away.
Okay, good morning, everyone. I don't see my video, but okay. Welcome to our symposium this morning. It's always exciting time for the instructors and our visitors to see what you've done with the results that you had. Slightly different formats since we're analyzing data from the past, but it's still going to be interesting to see what you did with that data, and also what other uses of barcoding you find. And it's very common, and I see it all the time now coming up. So I'm looking forward to each of your presentations. Like Nee said, we started this a while back to give experience to students who may not have the opportunity to do research and go through the whole process. So you started with a question, what is my fish? And is it named correctly? Then you gathered the data or saw how the data were generated to then analyze those data to answer your question. And now you've written an abstract, and today the final part is the presentation. So those are all the steps, except the final one, which of course would be writing a formal paper for peer review. But you've done almost all the steps of a science project, how scientists would do it in the lab in a standard place. And you might notice it's very different than how you were taught in high school about how the process of science. So I hope you've enjoyed it. It's always impressive to me to see students dedicate time, even though they're busy with classes and jobs and now probably more housework and things than we are used to doing during our academic years. And so it's very impressive to see you guys start with the project, stick it through, and I hope you found it to be something fun and different to take your mind off other issues. So with that, I'm ready to hear the talks.
Okay, thank you very much, Jim. So we actually have done, we've done quite a bit with this particular project, and we've expanded the amount of students and partners. With this particular year, we had about 70, up to 70 student participants. And even if we are at the UCR campus, 74, 72 is actually the physical capacity that we have amongst the 3 labs and the lab benches and space that we have. But regardless, during this particular time, we weren't sure, we weren't sure if we were still going to be offering this, and we're really glad to be able to offer this opportunity for you guys. So in any case, Jim did a great overview of what you guys had just accomplished. And in fact, we have a nice little video for you guys that will kind of encapsulate exactly what you had learned. And so I wanted to go ahead and show this as a start. And this way, we'll bring our presentation to a little summarization, and that way, you guys can see, hopefully you enjoy this little clip that we found. All right. Well, thank you very much. Okay. I was going to warn everybody, this was done by live real actors, and they put a lot of time into changing and creating that little barcoding debacle there. But yeah, essentially that would have been very interesting if you're able to learn a little bit of DNA barcoding through that particular incident. Gladly, nobody had drinking or eaten some random item and had something drastically change. So in any case, we're going to take a moment and I'll be adding all of the additional students for the first group. They're going to be jumping in and yeah, they'll be presenting, or we'll have our first presenter, the Moreno Valley group, and this is group number 3. So as I get everybody included here, it will just take a quick second. And again, so if you guys have any questions, we do ask that you either type in the chat or you can use the Q&A function, or again, you can raise your virtual hand and we will unmute you to ask it directly. All right. So it'll just be 1 second here, and let's get the rest of the group onto our stage.
Hello.
It'll be 1 moment. We're still adding everybody here.
Mm-hmm.
Okay, and it looks like we have 3 members of your group, and I just want to confirm that this is everybody. That's kind
One of them are
actually
not.
4. Okay. And I believe I have Samantha available too.
Yeah. Oh, sorry. I was going to say that I'm from the Mount Sac 1 group.
Oh, okay.
I'm from Mount Sac 1 as well.
Oh, I'm looking at
Yeah,
the
for
wrong
MVC,
group.
it's only me and Angela.
No worries. Then let's go ahead and put some of our panelists back.
I've been abducted.
All right. We'll put you back there. Sorry about that. Okay. Well, Janelle, Angela, thank you very much. You guys can take it away. Okay, Angela, your group, you guys are welcome to start anytime.
Hi, my name is Angela Marie Canaria.
We're sorry, we weren't hearing each other. Okay, I'm gonna start. Okay, my name is Janella Angeles.
My name is Angela Marie Canaria.
And we are from Moreno Valley College. So
This is our Sequence to Success DNA barcoding presentation.
Okay, so according to a 2019 report reported by National Geographic, more than 20% of fishes sold in restaurants and markets are mislabeled. So in this experiment, we are tasked to determine if the fishes we acquired from these stores are actually labeled correctly by using DNA barcoding.
DNA barcoding is a method used for rapid species identification based on DNA sequences. The sequence data are used to barcode the organism and to make distinction from other species.
Okay. Because of the low percentage of mislabeled fishes, Angela and I hypothesized that the fishes that we are to analyze are actually labeled correctly. So next slide, please. Okay, in this slide, we're gonna talk about the steps of DNA extraction.
Step 1 is to label the test tubes and add a small piece of fish sample. Step 2 is to crush the specimen and add buffer extraction to each tube, making sure to dispose of the micropipette tip before each use to avoid cross-contamination, and then grind the fish along with the extraction buffer.
Okay. Step 3 is to add 120 microliters of SDS. SDS is a detergent known to aid cell lysing. Step 4 is to heat the samples in a 65-degree heat block in order to separate the DNA from its cells, lipids, and proteins.
Step 5 is adding 225 microliters of potassium acetate. This will precipitate the SDS and the SDS-bound proteins to allow the removal from DNA. Step 6 is to ice the test tubes for 5 minutes and then centrifuge the tube for 5 minutes. This would separate the supernatant that includes the DNA from other cell materials.
Okay, transfer the supernatant to a new tube using a microfiber cloth in order to strain out the clumps of proteins that form. Next slide, please.
Continuing on DNA extraction.
Okay. Step 7 is to add 600 microliters of isopropanol. The alcohol would bind to the DNA, causing it to clump up together. Afterwards, we centrifuge it for 5 minutes.
Step 8 is removing the supernatant. After centrifuging, gradually pour out the supernatant and use a micropipette to remove the last drop. Step 9 is adding 500 microliters of 70% ice-cold ethanol and centrifuge it for 5 minutes to wash out the remaining SDS, salt, and other cell materials that's still left on the test tubes.
Step 10 is to pour off the ethanol alcohol. The following step is to heat the samples in order to evaporate the remaining alcohol. The last step of DNA extraction is to add 50 microliters of resuspension buffer in order to keep the DNA from degrading. Next slide, please. On this slide, we're going to discuss about PCR and gel electrophoresis.
PCR is used to amplify DNA sequences. It starts by heating targeted DNA at 95 degrees Celsius. This will denature it, separating into single strands. Afterwards, PCR lowers the temperature to 50 to 65 degrees Celsius, which allows the left and right primers to anneal to their complementary sequences. Following that step, the temperature is raised once again to 72 degrees Celsius, allowing Taq polymerase to bind to each primers and synthesize a new DNA strand. Denaturing, annealing, and expanding is one cycle of PCR.
Okay, now I'm going to discuss about gel electrophoresis. So it helps visualize the results of PCR. Electric current is applied to the gel in order for the negatively charged DNA to move towards the positively charged current. Because cells have different sizes, they would move in different speeds and will have different lengths. Seeing the different DNA bands will help us analyze the fish species.
Next slide, please.
Slide, please.
In this slide, we'll be talking about DNA sequencing. After amplifying the DNA, the results are sent to a DNA sequencing facility. Each student is assigned 2 fish samples to analyze. After determining the 2 fish samples assigned to us, we then use the DNA Subway website to analyze the fish DNA sequences. After watching provided video tutorials by Dr. Burnett, we were able to complete the DNA analysis. In the website, we were shown the trimmed version of our sample, the barcodes of our samples and species, and an evolutionary tree of the species that are closely related to each other.
Next slide, please. Okay, on this slide is our DNA analysis. Me and Angela analyzed 8 fish samples in total. And looking at the table, you can see the results of the DNA subway. We can see that in the table, the sockeye salmon, OPA, Dover sole, bonito, and tuna are the fishes that are labeled correctly. So based on those results, 5 out of 8 fishes that we analyzed were labeled correctly, and therefore it supports our hypothesis that fishes sold in markets are labeled correctly. Next slide, please.
I will be talking about application. What is another use for DNA barcoding? Another use for DNA barcoding is the conservation and protection of endangered species. By DNA barcoding, we can ecologically monitor species to keep track of their presence and absence in different regions of the world. If they are at risk of endangerment, then we can find out why. If it is because of the environment they are living in, if another species is maybe causing the endangerment, or if there is any presence of a disease that is known or unknown that is in their DNA that is causing them to decrease in certain rates.
Next slide, please. And that's the end of our presentation. We would like to say thank you to Nitran,
Dr. James Burnett,
Dr. Katie Burnett,
Dr. Chedi,
Alex Cortez,
our peer leader Devontae King,
and the rest of the research staff coordinator for this research opportunity.
once again, thank you for the wonderful experience, and we hope you all enjoyed our presentation.
That's the end of our presentation.
All right. Thank you very much, you two. Very, very great job. Oh, I was like, Angela, come back. So at this point, yeah, very, very good job. You guys are right on time too. So if anybody, do you guys happen to have any questions for Janella and Angela? Please go ahead. You can type in the chat or raise your virtual hand. And of course, we even have the chat feature here. And Dr. Burnett, Jim Burnett sends his regards and says, great job.
Thank you.
Yeah.
Thank you so much.
Okay. So the other Dr. Burnett, Dr. Katie Burnett, is asking, what was the most challenging part of this project?
It was probably communicating with the other group members. As you can see, there were like six of us in the MVC group 3, but only 2 of us participated. It's probably because they were too busy and stuff like that. But yeah, that was the most challenging part, to like get everyone together to participate.
All right. Yeah, glad you brought that up, because that's exactly one of the things that you will go through, and everybody goes through when it comes to this project, is it comes down to group work. And we understand it's challenging, and probably even more challenging that everybody's at home.
Yeah.
Okay.
What
Mr.
did we
Cortez.
enjoy?
Yeah.
Okay, okay, sorry.
Oh, no.
Sorry again. What did we enjoy most about the project? It was probably watching how the DNA extraction happened, because I'm taking biology right now and we're all just watching it virtually. We haven't really seen it in person yet. So it was really fun to like see the DNA extraction happen done by a real person. And we were able to like watch it again to really understand what was happening and stuff like that.
I enjoyed sequencing the DNA on the DNA subway.
Yeah.
That was like the fun part for me.
Okay, any additional questions for Angela and Janelle? All right. Well, thank you very much, you two. Really great job.
Thank you.
We appreciate it. And then hopefully you'll stick around till the end. We will see you then.
Thank you. Have a great
All right,
time.
you too. Okay. So just, and then we'll, yeah, you guys could turn off your video there. Awesome, thank you. All right, next group. Now you guys can turn on your videos as you get added along. Okay. All right, so our next group is going to be Mount SAC or Mount San Antonio College, Group 1. And your presentation is up. Something definitely smells fishy. So your time will start now. I'll let you guys take it away.
It won't let me turn on my camera since you turned off my camera. Thank you very much. Okay. Have you ever wondered if what you're eating is what you're is actually what you think it is? My name's Anson Lee, and I'm from Mount SAC Group 1.
I'm John.
I'm Liliana.
I'm Grace.
I'm Michael.
And I'm Samantha. So let's get right into it on the next slide. So I'll explain real quickly what DNA barcoding is. It is used for identifying species, not only fish, and from a short section of variable DNA gene, the ribosomal 16S gene in this case. And we align them and compare them to other DNA sequences on the database to identify what species it is. So let's get into talking why that is important on the next slide.
Thank you, Sam. So in 2018, Oceana conducted a study on the incidence of mislabeling in the United States. They did a study on 449 samples collected from 24 different states across the country. They found that an incredible 1 in 5 fish were mislabeled, mostly sea bass and snapper. And they've also found that 1 in 3 establishments mislabeled their seafood. The problems with species substitution such as this is that it undermines conservation efforts and puts vulnerable species at risk. It also is a health concern for the general public, as when they buy their fish, they need to know what it is, so that way they can have their health maintained. And on the next slide, we can talk about the methods.
Okay, thank you, Michael. So my name is Grace, and I will explain how we extracted the DNA from fish. So we took a piece of fish and ground it with a micropestle in a labeled test tube. We then added 120 microliters of SDS, which is basically an extraction buffer that helps denature the proteins. Then we placed it into a heating block and added 225 microliters of potassium acetate, which precipitates the SDS. This is put into ice and then centrifuged, and 700 microliters are put into a new tube. 600 microliters of isopropanol is then added, which clumps the DNA and centrifuged again. We decant the supernatant, then add 500 microliters of ethanol, which washes away the buffers. It is again centrifuged, and the tube is decanted and dried. We then add the resuspension buffer, which gives us our DNA sample. And in the next slide, Liliana will explain the process of PCR and bell electrophoresis.
Thanks, Grace. So for our polymerase chain reactions, a solution of MyTag PCR mix, primers, and water is distributed to 3 microtubes. Fish 1 DNA is placed in tube 1, fish 2 DNA in tube 2, and water in tube 3, which serves as our negative control. All tubes then go into the thermocycler so that reactions can take place. To visualize our PCR results, we use gel electrophoresis. A DNA ladder and each of our PCR samples are placed into individual lanes of our agarose gel. Then the gel is run and DNA bands are visualized using a UV light box. Next slide, please. Once our PCR samples are cleaned and sent for sequencing, we can use the FishBase website to determine the identity of our presumed fish and the DNA subway database by CyVerse to compare our sequences to those of other fish species. On the next slides, we will share with you the results of our experiment.
It's the next slide, please. So thank you, Liliana. And so in our results, we could see that only a third of the 12 fish samples were the species that were labeled as. And on the next slide, we can observe that the presumed barramundi turned out to be milkfish. So both samples turned out to be the same fish. And 2 rockfish samples were catfish. So basically, this just shows that majority of our fish samples were mislabeled. On the next slide, please.
So where does this DNA barcoding come from? It comes from this sample. So as you can see, there's lines, and each of the lines represents essentially a mismatch, a specific sequence of match or mismatch. So if you, will you click and highlight, please? So in this section, you'll notice that this is our Wahoo sample, and you'll notice only one difference between these two samples, and the one is that black line. That shows that there's one mismatch in the sequence that we were examining. So what this shows is that it increases the likelihood that this is the fish Wahoo. So bit score, which was what you saw earlier, is determined by the alignment length as well as the number of mismatches. Next, you'll be able to see a phylogenetic tree that was generated from these samples. The closer they are on the left is the time at which, left to right, you'll see that it's the time from when the species diverge. So if you look, can you highlight, please? This sample is of our tuna samples. You'll notice that they're all closely related to each other, more so than the other fish, since they all come from the same branching lines. An interesting fact about this is that it's even more related than the DNA Subways tuna sample. An interesting fact also was that out of all the fish, all the tuna were mislabeled as different fish. And this may be a product of bycatching. John will explain that.
Thank you, Anson. So as you can see on the next slide, so now that we know, we established that fish are more commonly mislabeled than we would think, the fish populations over time downsized due to human activities, such as the cowcut rockfish in Southern California, whose original population solely from overfishing and bycatching has been reduced to 4% of its original population. So on the side here, we just have another study by Oceana, and it's just a similar method of analyzing fish, and you can see that there are alarming percentages that are mislabeled. So with DNA barcoding's potential for identifying species, we can better regulate fisheries to avoid these harmful practices. Utilizing this technology more abundantly at early stages of the supply chain before reaching consumers would better protect consumer health while helping sustain our ecosystems. On the next slide, please. So reducing the decimation of these endangered fish, more fish can contribute to the ecosystem, such as food chains, for example, or even ecological cycles. So below we have a diagram. This is the nitrogen cycle, for example, where fish waste containing ammonia provides nourishment for plants that the fish eat. And so the cycle can continue, so forth. So in conclusion, DNA barcoding technology can contribute to better regulations that would benefit humans and the environment alike. Thank you.
Hey, thank you very much. Yeah, great presentation. Okay. All right. Does anybody happen to have any questions? Looks like Mr. Cortez, Alex has asked, what did you guys all enjoy most about this project?
I'll start. I did like doing the research, the background research, and the future application of DNA barcoding. I just found that was a really fun thing to look into, and just, really puts a real world application to this whole experiment.
Yeah, to me, probably just the experience itself and getting experience into research. To me, was really fun.
I actually liked my group a lot. We actually worked together, I think, pretty well. One of the main things I thought that was really interesting was when we were actually seeing the professors do all the experiments. That was, for me, the most interesting thing, because I want to do research, and actually seeing like the wet work done, that was really, really fun.
Okay. Well, if I can comment, I thought you guys did an amazing job. Thanks for the transitions. Were great. We're spot on. I think that really shows from the practice session yesterday to how you put it together and work together as a team now. I'm also very glad you guys all came looking really good. You guys all dressed up, you put on your makeup. I took like at least 3 hours to get ready this morning, so if you could tell. And yeah, I really appreciate you guys putting that effort in. So thank you very much. You guys should definitely be proud of yourself.
Thank you.
Thank you.
Okay.
Thank you.
All right. Now, if you could do me a favor and just turn off your cameras, and we're going to bring on the next group here.
Okay, perfect.
Thank you.
All right. Thank you. So we're going to slowly get the remaining or the next group coming on over here. And it looks like we have a couple. Okay. And then as the next group does come on in, you guys are welcome to turn on your camera so we know that you are alive and well. Okay. Thank you very much. And I believe we got your group. I believe we got the entire group here. So without further ado, your presentation is up. Again, unfortunately, it's not the most recent one, so it is whatever you had before here. And you guys are welcome to take it away.
All right. Okay, guys, so we are ECC2 and we are doing 16S Jump Street, and we'll just get right into it. So if you can go to the next slide, please.
Hello, my name is Natalie Otsap, and I'm here to talk to you guys about the background of this study. So mislabeling fish is actually a very big issue for consumers. A nationwide study was conducted with 1,200 samples of fish from over 600 retail stores, and it was found that 33% of all seafood are mislabeled, and it's actually even higher in California at 52%. The problem with mislabeling is that it can cause cheaper fish to be mislabeled as expensive and vice versa. Not all mislabeling is intentional though. Some species of fish are misidentified because of their similar physical appearances. Mislabeling also poses environmental and health risk. Specifically, some fish have higher levels of mercury than others, and consumers who want to support their local fishermen are sometimes served products from foreign countries. Next slide, please. Next slide, please. Oh, thank you. So to solve this problem comes DNA barcoding, which is a method that takes short DNA sequencing from a specific gene of each sample, and it's taken and compared to an existing database of DNA sequences, and that can give us the most accurate identification of any organism. This works better by looking at the genetic level rather than relying on physical attributes for identification. We believe that this method will better detect food fraud and hopefully reinforce security amongst consumers by providing the proper labels for fish they buy. We use 16S rRNA to analyze our samples and then amplified them with PCR. Next slide, please. And I got it handed off to my colleague Quang, who will explain in further detail the method. Next slide.
Hi, so before we can do the PCR, we have to isolate the genetic material from the cells, and we do it physically at first by using a micropestle and then grind the sample thoroughly. Then we can do it chemically by using SDS, and SDS can bind into the protein, the lipids, and denature it. The next step is to remove the SDS and the SDS bound protein by adding potassium acetate. And then we can filter out the top part, which is the supernatant that include our DNA. And we can adding isopropanol into the sample to precipitate our DNA. In the next slide, next slide, please. Yes, in the next slide, we adding ethanol as a wash alcohol to remove all the impurity within the test tube, and then we resuspend it in a centrifuge, and then we are done. And the PCR step is actually very straightforward. We're adding primer, the forward one and the reverse one, and we're adding a mixture that containing our DNA polymerase, the building blocks and the buffer, water, and our DNA samples. We are making 3 test tubes. The first one including our first fish, the second one including our second fish DNA, and just for quality control, we have a third test tube with just water and all the stuff mentioned above, and we can replicate the target section of DNA multiple times by PCR. And one way we can make sure this process works is in our next slide. Next slide, please. Yes, we do electrophoresis, which is just a quality control test to check, if the DNA has been amplified, the length of the DNA section, and that's it. And we send the rest of our DNA to the computer to do analysis in our next slide.
Thank you, Quang. So now here we have a quick chart detailing all of the samples that each of us sequence, as well as certain characteristics. And we'll go over the details a little bit later on. Next slide. Here we also go more into detail about the different fishes that we had. For example, we had supposed smelt, mackerel, swordfish, and so on. Next slide. And now to the results. On the right-hand side, we have a DNA-based phylogenetic tree that compares the genetic relationship between each fish sample and their respective species identity. The highlighted names show the presumed identity of each sample along with its genus that it truly belongs to. The color green indicates the samples that match their original label. The color red is for a mismatch, and blue is for inconclusive, but we'll get to that in a bit. An example of a match includes one of the fish samples that was presumed to be fresh rockfish, and DNA analysis showed that in fact, it belonged to the genus Sebastis, which includes all rockfish species, meaning it matches labeling. In particular, its species identity is most likely to be Sebastis entomalis, also known as widow rockfish. An example of a mismatch includes a fish whose presumed identity was pollock, but in reality, it was identified as belonging to the genus Dysosticus, which includes all species of toothfish, and toothfish are not pollock. Next slide. To summarize, out of all of 12 fish samples that were analyzed, only 5 matched their labeling, 5 mismatched, and 2 were inconclusive. Now to elaborate why there were inconclusive results. So for one of the fish samples, the presumed identity of the fish was unknown, which did not allow for a proper species comparison. For the second fish sample, we identified its DNA as that of a bacterium. And since fish are not bacterium, this highly suggests that the sample was contaminated and therefore it could not be compared. Next slide.
All right, guys, so I'm going to be taking over. My name is Natalie Liberian, and I wanted to go into other applications and how we actually already use PCR and DNA barcoding. So to start off, we know, what is it called? The model organisms. Model organisms are organisms that we study as scientists and while doing research that can later help humans and us with our research for modern medicine and other ways. So for example, we have a primitive fish here on the left. It's called the horseshoe crab. It's been around 445 million years. We actually harvest their blood because we found from studying their DNA that they have a very sensitive and advanced immune system. So we now use their blood, harvest it to use it to make sure that our injections that we use in hospitals are not contaminated. Also, E. coli, it's eaten by the C. elegans, which is the roundworm shown on the right. And we now study that as a model organism using their DNA to test how humans can better coexist with bacteria. And the way I thought was in the middle there, you have a diver holding a sea cucumber. So I thought, what if we continue to study, short strands of DNA of sea cucumbers and compare them to humans, we can find differentiations, see which ones have to do with regeneration, and maybe we can simulate regenerations in humans, which sounds crazy, but for those who suffer from limb loss or nerve damage, we can somehow use that, what we learned from our research, and use it on humans with sea cucumbers. So humans are smart. I think we can do it. So I'll pass it on to the next slide, please, for Andrea.
Okay, hi. Okay, so from PCR and DNA Subway, we learned that out of the 12 fish samples, only 5 were labeled correctly. So, and the other 5 were mislabeled and the other 2 were inconclusive. So taking in the 5 fishes that were mislabeled, we put in the clades over here, as you can see in the figures over there. And then, for example, we have figure 1, swordfish, and then Coryphena hippus, it's actually mahi-mahi. And so they're part of the same clade, they're sister taxa, and so they share physical characteristics. So based from these, we have a strong leading evidence that mislabeling may have occurred due to actual confusion in the physical characteristics of fish because they are closely related. So next slide, please. Okay, so in order to combat fish fraud, we recommend to use DNA barcoding, although this has limitations because it can be quite expensive and it's not that available for small owners. Plus, it's laborious and a taxing effort if you have to barcode multiple fishes at the same time. Although it would present a nice and more accurate picture because you actually get to look at the DNA of the fishes. And so we also recommend that fish products be tracked from the shipment until they reach the actual destination so that we can trace the products and then provide also accountability in case mislabeling does happen at the supply chain, which is what the Bill Safety and Fraud Enforcement for Seafood Act is all about. Next slide, please. Next slide. Okay, thank you. So what we have learned from this is that mislabeling does not only pose environmental and health risks, but also economic losses, as seen in the figure over here. Consumers could be paying more for less if you get substituted fish. And so for us, what we can do for now is to ask, is the fish farmed or wild? When, where, and how was the fish caught? The more knowledge we have and transparency will allow us to make more informed decisions. And that's it. So next slide. We would like to thank Dr. Darcy McClellan, Nitran, the whole UCR CNAS Barcoding Challenge team, Dr. James Burnett, Dr. Katie Burnett, and Mr. Alex Cortez. Thank you.
All right, thank you very much. Great presentation, everybody. All right, does anybody happen to have any questions? Looks like Mr. Cortez did ask. Yeah, standard question. What did you guys enjoy most about the project?
I personally enjoyed using the DNA subway. That was so much fun. Like, it's something that I think someone from another group mentioned this, but I also want to get into research. And that just really made me feel like a scientist for a day or two. So it was nice.
Yeah, personally, I enjoyed DNA Subway too, and researching about the whole fish fraud and mislabeling issues. Because I really didn't know about that, and so it's great to be more informed about what is actually happening, and that sometimes it's not really intentional. It's actually just confusion. Like most of us will actually commit like honest mistakes.
I also have to agree with Andrea. That was my favorite part, was knowing about this issue and kind of how easy it is to solve with DNA barcoding. I mean, wow, that's super cool.
To answer the question, what did you find surprising in your project? For me personally, what I found surprising was that even with the use of DNA subway, we were able to really identify the genus of a certain fish sample, and we had a high probability of what we thought the species was, but there's no guarantee. And I thought that was what was most interesting, because it just goes to show that even with the most advanced science, there's no real guarantee anywhere, just our best estimate.
Yeah, and plus what's surprising is that, that 33 over like half of the fishes are mislabeled, especially here in Southern California. And so when I saw those figures, I was, wow, like, wow, I could be actually paying more for less.
Well, I found
Specifically,
surprising.
sorry,
Oh, sorry.
go Quang.
Okay. What I found surprising is that we can do this online in virtual meetings, and we can do a proper and correct research. It is very satisfying feeling I having right now. Yeah. After we
All
finish.
right.
All right.
Thank you very much. And yeah, you guys all hit it spot on. Quang mentioning about research online. Yeah, I mean, that's one of the good things. One thing to keep in mind is that you're able to do this because Dr. Burnett, Alex, and Dr. Chetty had all done this experiment for you live in a lab. And they also had to send out samples to get sequencing. So there's still a physical component to doing the work, but at the same time, a large component and the most time-consuming component, arguably, is going to be the analysis that you guys did. And I also loved one of your last slides for the conclusions, illustrating the fish. Even though they're identical pictures, it's very easy to see if you walk into a grocery store and a restaurant, how do you know what you're getting? And you can see that there are clear financial benefits or disadvantages if you don't know what you're getting. And yeah, really great job, everybody.
Thank you.
All
Thank
right,
you.
Thank you so much.
Thank you.
you. All right, now may I ask you guys to turn off your cameras? We have our next group already on board, and the next group can turn on their cameras. And I'm going to go ahead and change the view here. Great. All right, your presentation is already on board. You guys may take it away.
All right, good morning, everyone. We're Mountside Group 2. My name is Michael, and my group members are Dima.
I'm Dima.
My name is Jillian.
I'm Tiffany.
And my name is Jeffrey, by the way.
I don't know what just happened. I just got kicked off. Okay. And our presentation was titled The Significance of DNA Barcoding. Next slide, please. So fish mislabeling is still a widespread issue. There are many reported cases of fish fraud, the phenomenon where you buy an advertised species of fish, but you just get a different one. Based on the National Seafood Fraud Testing conducted by Oceana, summarized by the map shown here, there are a significant amount of fish mislabeling cases throughout the US. And as you can see circled in the black down in the bottom left, here in Southern California where we live, more than half of the fish that were tested, around 52%, were of a mislabeled species, which is just quite concerning. Next slide. So why is it important for fish to be identified correctly? Well, for obvious reasons, the significance lies in the contents of the fish. Many people choose to, or are required to, consume certain types of fish for its nutritional value. Fish are known to contain these omega-3 fatty acids, of which EPA and DHA are the two main kinds. As shown by this table on the right, mackerel and salmon contain the highest concentration of these omega-3 fatty acids, whereas sea bass, located fourth from the bottom, contains one of the lowest concentrations. So just imagine if you walk into your Albertsons and you were specifically buying mackerel to supplement your diet, but you're just getting sea bass instead. This to me at least is a serious health risk. Next slide, please.
Also, serious allergic reaction to certain fish can cause death. Some fish contain high levels of mercury, which is toxic. If fish are misidentified, it can put pregnant women at risk. The picture on the side demonstrates that swordfish has the highest level of mercury, along with sharks. On the next slide please. There are 2 fish samples given in order to find out whether these are properly labeled in stores and restaurants. The fish was cut into a small piece of tissue and was placed in the tubes. Extraction buffer was added to the tube. This would open up the cells, and after that, the tissue was ground. 10% SDS were then added. SDS are used for cell lysis. Once that's done, the tubes were placed in bedlock for at least 20 minutes. At this point, the nuclei and mitochondria had been lysed with solution. In order to precipitate, CoAC was added. CoAC is acidic that would make the lipids less soluble and cause it to come out easily of the solution. The tubes were placed in ice for 5 minutes and then they were placed in the centrifuge. As you can see, 2 layers were formed. The bottom portion is referred to as pellet. This portion contained the cellular proteins and lipids, while the top portion was referred to as supernatant. And this contained the DNA. The supernatant was transferred into into a new tube using a microclot funnel. Next slide, please.
So after transferring the supernatant, we add isopropanol. What it does that it causes the nucleic acid to precipitate down the tube, and also it changes the water concentration. Then we centrifuge for 5 minutes. We will notice that a glassy pellet formed in the bottom of the tube. So we remove the supernatant and then we keep the pellet. We add iced ethanol on the pellet so we can rinse out the DNA and it removes all impurities. Then we centrifuge for another 5 minutes. We pour off the ethanol and then we add the resuspension buffer after we make sure that the ethanol has evaporated, and then the DNA is ready for the experiment. Next slide, please. So after that is the PCR. The purpose of PCR is to amplify the 16S gene. So first we have to prepare a solution containing our DNA, the primers, DNA polymerase, and NDTP, the building blocks. It'll go through many cycles and through 3 steps. The first step is the denaturation steps, where the DNA template comes apart. It occurs at 94 degrees Celsius. And then the annealing step, where the forward and reverse primers attach the DNA. And then the extension step occurs at 72 degrees Celsius. The DNTP attach the DNA by the DNA polymerase. After that, we use that gel electrophoresis to make sure that we have a product and to test the quality of the DNA that we have. Then we pick the best DNA sample and then we sequence it. After we got our sequence, we use DNA subway just to check what type of fish we had and if it matches or not with that given type of fish. Next slide, please.
So the results can be analyzed using DNA Subway. The first tool that you'll find in the DNA Subway is the sequence viewer. Next slide, please. Which allows you to visualize the sequence you produce from the experiment. And as you can see in the orange box, the sequence isn't clean and has a lot of noise at the beginning. And there's also noise at the end, but it's not shown in this picture. Next slide, please. And in order to clean up that noise, we have the sequence trimmer, which reduces the noise and the peaks we saw earlier and cleans up the sequence, as you can see in the photo now. And once the sequence is clean, because this is a fish sequence, it should be around 600 base pairs. Next slide, please. After we get the clean sequence, we use BLAST-N, which is a tool that aligns nucleotide sequences to find a long stream of bases with little to no mismatches, so it's more likely to be the correct fish species. Next slide, please. But there are problems that happen, like low quality score, which is where the DNA is damaged, and this can lead to no or completely incorrect BLAST-N results. And in this case, the results will be deemed inconclusive. Next slide, please.
But when you do get the proper sequences from BLAST-N, we'll use muscle to compare them, and this tool will let us visualize where the discrepancy lies. It can also be changed to ATCG view, where you can see the exact location of the mismatches and matches of the sequence that are chosen to be compared. Next slide, please. And lastly, we'll use a phylogenetic tree where we can identify the most closely related sequences and determine the identity of the fish. The less nodes, a node is where the arrow is, the more closely related the sequences are. In my case, the sample inside of the orange box matches the sequence of an Atlantic cod when my fish was labeled a Pacific cod. This would deem my sample a mismatch. Next slide, please.
So in total, we have sampled around 12 fish, and out of the 12 fish, only 3 of them are actually labeled correctly. If you look closely, I put a happy face on the chart to designate which fish are actually correctly labeled. And also notice how these 3 fish, the one with a happy face, has a really high bit score in comparison to the other fish. In addition, if you look on the chart, you see a line on that purple line over there, that's the E-spec value. That is a degree of errors that BLAST-N may have made in trying to correctly identify the fish. And E, nevertheless, it's still not the same fish we expect it to be unless it has a really high bit score. And if you could see on the last 2 fish, we were not able to run it through BLAST-N due to the damage of the DNA mentioned earlier. Next slide. In total, we have 7 mislabeled fish, 2 unknown and 3 actually correctly labeled, which means we may be eating the wrong kind of fish 3/4 of the time. And next slide.
So now, another use of DNA barcoding is to identify disease vectors like mosquitoes, which are known to spread diseases through the bite. So it is important to know what mosquito species can carry a certain disease so it can be prevented. In the next slide, thank you. As you can see, it is easier to differentiate a mature mosquito from a pupa or larva, which is where DNA barcoding plays a big role in identifying the species at an earlier stage. Next slide.
So in the same line of disease prevention, another application of DNA barcoding is for foodborne illness prevention. According to the CDC, there were 16 outbreaks of foodborne illness in 2019 and 7 outbreaks of foodborne illness in 2020 so far. You might remember the E. coli O26 outbreak that happened at Chipotle in 2018, which got a couple people hospitalized and forced some Chipotle locations to close down. And with DNA barcoding, we may be able to detect these pesky pathogens before they get to the plates of our consumers. Imagine if you're sequencing some red onions and you get some salmonella genetic material back instead. You might be surprised to find out that this salmonella in red onion outbreak occurred in August of this year, fairly recent. And we're just hoping that this aspect of DNA barcoding can be used to prevent similar outbreaks in the near future. And in any case, we would like to thank everyone for watching our presentation. And we would also like to thank all of the people that made this opportunity possible. And we look forward to the rest of the symposium. Thank you.
Okay, thank you very much, everybody. Very good presentation. I will echo Alex's sentiment. Nicely done. And yeah, very good job of very thorough analysis and making all the charts and really clearly laying out exactly your results as well as how it applies. So really good job. Okay. And Alex would like to know, what did you guys enjoy most about your experience or your project?
Well, for my part, it's actually through making the table for the BLAST-N results, just because I wasn't really like understanding it very thoroughly what each of those means, such as alignment length, bit length, and the E portion. And so I actually had to look it up, and I actually went into the area of bioinformatics, which I didn't expect to go there. But apparently I learned a lot of new things just by actually just taking the data and trying to find what it means and trying to find correlation to each of what we are talking about.
I'd say for my part, I would say that it would be the overall experience given the current situation we are right now. I'm sure all of us learned a lot from this opportunity that would potentially help us all in the future.
For me, my favorite part was how during the time when this started, I was actually learning about this in my biology class. So it was really interesting to apply what I knew from bio to this opportunity and what I learned from this opportunity to my bio class.
And for me, I guess overall, I just had fun. And the learning process was great. Like honestly, I just had so much fun learning about this. And I have this phrase. It's like you can go to the most fun places, but if you go with the wrong people, it's not fun. But you can go to the middle of the desert, and if you go with the right people, it's fun. So I had fun with my group. Yeah, it was just a great experience. Thank you.
All right. Thank you very much. And another compliment. You guys all look great. Really impressed. Jeffrey putting on the shirt and tie, hanging outside. Need to do some DNA barcoding on those dogs. They're probably all Chihuahua. But yeah, regardless, you guys did a great job. Again, appreciate the amount of work that you guys put in. So we will move on to our last group before the break. Running a little bit late, but that's okay. You guys are spending the Saturday morning. So I would like to ask the Mount SAC group 2. You guys can turn off your cameras. And then with the RCC Norco group 1, I believe we have a couple attendees or a couple presenters. You guys may turn on your cameras. Alberto and I believe Alexa. Great. And I just want to make sure, was there anybody else you were expecting from your group?
Yes.
Yeah, we're expecting Patricia, Myra, and Jason.
Yeah.
Okay, just verifying. Did not actually quite see them here. And I do have a hand up for Nathan. Nathan, would you like to say anything? I have unmuted you. Sure. Yeah, and if anybody else is part of this group, if you can raise your hand, you are welcome, and then that way I could add you on. Okay. I have a feeling I know what happened here, so I'm going to go ahead and add a few other members here. And then if you're part of the group, feel free, turn on your camera. All right. There we go.
I think
Yes.
one of them isn't part of our group, Gina. Oh, sorry.
No worries. Okay, well, your presentation is up, and I will disappear into the background. You guys can take it away.
Good morning, everyone. Alberto, Jason, Myra, Patricia, and myself, with the help of our peer lead, Nathan, have worked together these past 3 weeks to figure out how important DNA barcoding is in a simple everyday situation, like trying to figure out what you're eating when you go to a restaurant or when you purchase food from a market. Together, we've identified the species name of the samples we were each provided, analyzed our data, and concluded that most of the time, the food we eat, in this case, the fish we eat, is not what it's advertised to be. So I'm going to go ahead and pass it over to Myra.
Hi, everyone. Next slide, please. Where were we doing the abstract?
Next slide.
Next slide. Background. DNA background is a method used in differentiating species by studying the short sections of DNA form a specific sequence of genes. Earlier techniques of DNA barcoding was used by Carl Richard Royce to discover the archaea domain of life. In 1977, the modern method of DNA barcoding was proposed in paper by Paul D. and Herbert et al. In 2003, Herbert and et al. Had used voices techniques to identify arthropods in the 1980s.
Next slide, please. Fishing for answers. What are we attempting to learn about by studying the DNA sequence in the different species? What advantage does DNA barcoding have when determining the differences between species? How can we discuss a barcoding technique to differentiate between different species from an unknown sample? Is there a possibility for DNA barcoding to become a useful tool in our everyday lives? Next slide, please.
Hi, Alberto Munoz, and I'll be going over the experimental steps. Here we just have a flow chart of those steps. It's mechanical chemical DNA extraction, PCR, polymerase chain reaction, and gel electrophoresis. Next slide, please. So just a quick overview of the experimental method per step. For DNA extraction, we're going to extract and collect the DNA from the sample, in this case, the fish. We will break down the cell membrane in order to expose the DNA. We will use a centrifuge in order to separate the DNA from the debris, and then we will purify the solution. Next, through PCR amplification, we'll denature the double strand of the DNA by heating it, allowing the primers to bind. The DNA polymerase polymerizes a new complementary strand. We'll then lower the temperatures for the annealing step of the reaction. We will rapidly heat the DNA in the elongation step. We will go through these 3 steps about 40 times over. The number of the sequences will grow exponentially. So by the end of that, you'll have millions of identical strands, sequences. And the final step would be the gel electrophoresis. We'll prepare the solution by adding a basic blue dye. We will prepare the Argos gel, which our sample will go through. We'll pour the solution to the casting tray along with our samples. We will turn on the electric current. Since our samples have been coated with a negative charge, they will then move towards the positive end. At the end, we'll study the quality of the duplicated DNA sequence. And then as far as the serial dilution, so we could dilute and just have a sample that's not as concentrated to verify if how accurate our samples and experiment was. Next slide.
So for our results, 54.5% of the group's fish had been labeled incorrectly. 39.4% of the group's fish had been labeled correctly. And we did have a single inconclusive sample. I believe that we had it replaced with a second sample, but that did add a 9.1% rate of inconclusiveness to the data. Next slide, please. So our total sample count was 11 samples, including that inconclusive sample. 6 were incorrect and 4 were correct. Next slide, please. So here on the phylogenetic tree, you can see that the correctly labeled fish at the top, Myra's fish number 2, Alberto's fish number 1, Jason's fish number 1. In the middle, there's that whole section of branches where all of our fishes are labeled within the same branches. I believe that that means that we have closely related ancestors, but they were all labeled within the same branches. I'll go ahead and pass it over to Patricia. Next slide, please.
So I'll be presenting the discussion, and I'm under Nathan for some reason, and I don't know how to change my name, so excuse that. So the methodology used in this research project reflected the salience and the importance of using science to identify any errors that we can find that could possibly interfere with the correctness of scientific results. In conclusion, the 16S barcoding gene was useful for finding what type of fish the sample really is. The identification of fish species was efficiently demonstrated using DNA barcoding. Finally, we can presume that sequencing is an effective method to distinguish fish species.
For the conclusion, we were discussing what was the reason for fish being mislabeled on a regular basis, because a large percentage of fish has been mislabeled or either accidentally or on purpose. But either way, the results of mislabeling of fish can cause different health issues, such as high mercury content or just people being allergic to that certain type of fish. Usually, if it's accidentally mislabeled, it's either because fishermen have a big haul, so they can't take their time individually carefully looking at each fish. So they just make a quick guess or quick analysis. They analyze it quick. I'm sorry. The other one is it's either purposefully mislabeled because usually, or if the fish that they caught were not of high quality, they would try to make it, they pass it off as a higher quality fish. So the FDA lists acceptable labels for seafood, but there are also vernacular names that are not accepted by the FDA. These vernacular names also allow room for fish to be mislabeled. And I am. For the alternate use for DNA barcoding, I came with a question: how much of the world's food has been genetically modified? A majority of the world's population doesn't really know what exactly they're putting in their bodies. Many types of foods that state are GMO free can almost be guaranteed to be false. GMOs, the reason we modify foods is either for fertility for a higher yield, or infertility to make them infertile if someone gets their hands on those seeds. It's intended to increase the nutritional value of farmers' crops. But there have been many, even the organic crops grown can contain genetically modified seeds. So we use the DNA barcoding GMOs are just it's a broad name given to what's actually been done to organisms. So maybe we use DNA barcoding to figure out exactly what has been done to those organisms. Next slide, please. I would also like to give thanks to Dr. James Burnett, Mr. Alejandro Cortez, Dr. Katie Burnett, Mr. Netran, and our peer lead, Nathan. Unfortunately, we forgot to add Dr. Chetty, which we would also like to thank her for performing the lab for us. And that's it.
All right, thank you very much, everybody. Let me bring everybody back to the screen. Okay, well, we finally got you all in here, and I think we figured out the problem. That was one of the reasons why we did request everyone register on your own. Don't share links, because your name will pop up as the person who shared that link. It is a unique link, but it's okay. So if you did happen to get in here, you are capable of changing your name. So you got the sign of approval from Dr. Herrick. He says, great job to you guys. All right. And the real Nathan looks like he says great job as well. So does anybody have any questions for the group? And again, you guys are welcome to type into the chat. You can raise your virtual hand. You can even attempt to use this Q&A function, but I think the chat would probably be best. Okay.
I think I saw that Alex asked us what we enjoyed the most about the project. And I would say that I enjoyed the most like the research and getting to see, unfortunately not in person, but through Dr. Chetty and the rest of everyone who did the project, how the DNA extraction happens and how we get to the point of finding the species of the sample that we analyzed.
Same. It was a nice experience being outside of class. You don't communicate with too many students. So, you know, being put in a group with the fellow students who are in bio, I thought that was a really great experience to be able to discuss biology in a group, even though we can't be on campus. So I really appreciated that.
All right. Looks like Dr. Katie Burnett did ask, what advice would you like to give to the next group of students who would like to try this project?
I would say go for it. It's a new experience that doesn't really get offered much, especially if you're a STEM major. Since I entered community college, I never specifically joined anything or did anything like this. So it was a new experience for me. So it was nice.
And like, if you feel like you have no experience in something like a research project, like join anyway, because it's something you can learn, something new to try. And at the end of the day, you will learn a lot more than what you started with.
Okay,
I would
well,
definitely,
go for it.
I would definitely join. I mean, it was my first time as well, and I really enjoyed it. It was interesting.
I'd say it's a great opportunity to dive into research and just learn more about basically biology and DNA, even if you've never had any past experience. Like this was my first time, and I learned a lot through this process, and I'm so thankful.
Okay. Well, thank you very much, everybody. We will begin a 10-minute break. We are running a little bit late, but again, that's okay. It's for you guys, and we're enjoying all of your presentations. So the timer has started. Everyone can go ahead and go have a quick coffee break and restroom, grab a snack. And yes, we can hear the music. We will see you back here in a little bit less than 9.5 minutes. So thank you very much. Okay, welcome back. That was a much needed quick break, but yeah, I'm actually ready to hear the second half of our presentations today. So our next group is going to be El Camino group number 1. And yes, if you guys may all turn on your cameras, that would be amazing. Okay, your presentation is already uploaded and you guys may take it away.
Okay. Hi, everyone. My name is Hannah, and in my group is Eric, Murnaz, Donvi, Jasmine, and Jedediah, and we are from El Camino College. Our presentation is called Something Seems Fishy, and I'm going to pass it off to Eric to give you some background about what we did.
All right, next. We will begin with the background of our experiment. So what is DNA barcoding? It is the process of species identification by using a section of DNA in a gene sequence. We can identify an organism by comparing it to a library of DNA sequences. We will be using the mitochondrial 16S ribosomal subunit as it is useful for identifying species-specific marks. Next slide. Why are we applying this to fishes? The biodiversity in fishes makes them hard to identify. There are over 32,000 fish species, and restaurants may occasionally mislabel fishes for monetary reasons or lack of knowledge. So take a look at these two pictures. They are the same except for how they were labeled. Restaurants can increase the amount they charge by just saying it's a different type of fish. We will be using DNA barcoding to decide if the fish we were given were labeled correctly. And I'm going to hand it over to Murnaz. Next slide.
Murnaz, you're muted.
Sorry about that. Thank you. So as Eric said, DNA barcoding is a new method to identify any species based on extracting DNA sequence from a tiny tissue sample of an organism. First, we isolate the macromolecules such as proteins and lipids from fish DNA. Then by adding alcohol, ethanol in this case, to the solution, the DNA precipitates out of the solution. Next slide, please. The next step that we have to do is amplifying the target DNA barcode region using PCR. The PCR amplification has to work so that there is enough DNA for sequencing, and it also makes it possible to see. Next slide, please. DNA samples are loaded into wells of gel electrophoresis to separate DNA fragments according to their size. And also the purpose of doing this is to make sure what we did was right. Next slide, please. Now we have to determine the nucleotide sequence of the DNA. And the gel was used to confirm which samples can be sequenced. Next slide, please. In the very last step, we have to match the sequence to the database to find the matching species. Next slide, please.
Okay, so these are the results. We had 12 fish samples, and out of those 12, 5 matched their presumed species, 6 were mismatched, and 1 was unknown from the start. Okay, next slide, please. So these are the 5 matched species. Sorry, I don't know why it looks kind of weird on here, but and even though these were all a match, for some of them, we were able to get more specific results instead of just the general fish name. For example, we found out that the mackerel was actually an Atlantic mackerel, the catfish was a channel catfish, and so on. Next slide, please. So these are our 6 mismatched species. The coho salmon was actually masu salmon. The rockfish was actually a striped catfish. The manta ray was actually a pale-edged stingray. The Dover sole was a widow rockfish. The barramundi was a milkfish. And the silver side was the Pacific cod. So out of our 12 samples, 50% were mismatched. Some of these were conditionally matched, but Donvi is going to discuss those in more detail in a bit. Next slide, please.
Lastly, this was our unknown species, and we found out it was a blue rockfish. We had no information for this sample, but DNA barcoding allowed us to determine the species of fish with just a tiny amount of sample. Now I'm going to pass it on to Donvi for a discussion of the results.
So when looking at the overall results, 41.7% match while 50% mismatch. One sample had to be omitted because it had no presumed identity. There are several reasons as to why samples are misidentified. These include actual mislabeling, which might be done to reduce the costs of exporting or importing fish, or to make a larger profit from the said fish. And then there's accidental mislabeling, where samples may be mixed up in the lab, or the person labeling might not have enough experience to tell one fish from another. And also contamination may be a factor as well. Next slide, please. Out of the 6 mismatched samples, 2 of them conditionally matched. These samples are the Masu salmon, which was presumed coho salmon, and the pale edge stingray, which was presumed manta ray. Their real identities are part of the same class to the presumed identity, but they are different at a more specific level of classification. Next slide, please.
Through DNA barcoding, it was determined whether the advertised name of the fish bought matches its molecular identity. The DNA subway was used to run the data and get the blasted name of the fish. This way we determined if the fish species matched or not. Next slide, please. Information on some of the fish was incomplete. Some of the fish's names matched partially, and there was an unknown species. In this experiment, it was found that 50% of the species analyzed did not match the advertised name. This could be due to false advertising, experimental and/or human errors. Because of the high percentage of mismatch, possible errors, and incomplete information, the experiment should be rerun to verify results. Next slide, please. Jedediah will be talking about other applications for DNA barcoding.
Yeah, thank you, Jasmine. Another use of DNA barcoding. DNA barcoding can be used to identify type 1 diabetes in embryos before an implantation. As a reminder, type 1 diabetes is hereditary, and it occurs when the body's immune system attacks the insulin-producing cell, which is the beta cell in the pancreas. The risk of developing type 1 diabetes is increased by specific genes that provide instrument-making proteins that plays critical role in the immune system. But with DNA barcoding and other bioengineering techniques, as CRISPR-Cas9, this gene can be edited in an embryo before implantation. Next slide, please. How can this be done? So through in vitro fertilization, egg and sperm will be manually combined in the laboratory. DNA will be extracted from an embryo. Using PCR, we will amplify the DNA. The gene will be transferred to a gel electrophoresis, and the sequence, and we'll have a specific sequence to be targeted. The DNA will be compared to individual with type 1 diabetes, just like the parents, and the specific match will give us the gene which we need to target. If the specific match with this disease, then we can use CRISPR technique to correct and edit the genes. Next slide, please.
And this concludes our presentation. Before we go, we would like to give thanks to all the instructors and advisors for allowing us the opportunity to participate in this research experience. Thank you.
Okay, thank you very much, everybody. That was absolutely a great presentation and a lot of great organization. I appreciate everyone showing up on time. You guys all look amazing. And we will open the floor up for any questions. Mr. Cortez did ask, what did you find most enjoyable?
I feel like for me, I had 2 favorite parts. I really enjoyed using DNA Subway and actually being able to like put in the information and like find the matches. And I also really enjoyed working with my group. I feel like we had really good communication throughout it all, always group texting ideas and meeting on Zoom. And I think it was nice to have interaction with people who are not necessarily in our household at this time. And it was just really fun. This was my first research experience that I have done ever. And what I really enjoyed doing this research is, first of all, learning from all knowledge professors, and I really appreciate you guys. And the second thing was how it challenged my mind, and just to be able to think outside the box and come up with new ideas, like how it applies to real life, how we can relate science and what we learn in biology to what's actually happening around the world and solve humanitarian issues or diseases. So it was pretty fun and cool.
This was also my first research experience, so it was a really great opportunity to have.
It was amazing working with this team as well, and I really appreciate everyone and the efforts that they put. I want to thank especially the UCR for making it possible, and I learned a lot, and I'm really grateful, and I wish to use this knowledge to help the world tomorrow.
For me,
oh, go for it.
I said for me personally, it was a very real world opportunity, what we're applying the sciences that we learned in class to what we would do out in the real world. So I really enjoyed this experience.
For me, I think my favorite part about the project was just being able to work with my team and being a part of the whole process. And it was, my favorite part was probably when we, I think it was the first time we met like as a group, but we just got so much done and everything felt like so easy to do.
All right, thank you very much, El Camino team. Again, we always appreciate having students from El Camino join our program all the way out in Compton area. I know it's a far away from UCR, but we'd love to have you all on campus at some point, hopefully in the near future, and perhaps even in whenever you decide to transfer. Regardless, you guys did a great job. Really proud of you guys, and we will be moving on. So may I ask El Camino, you guys can turn off your cameras, and the next group we have is going to be RCC Norco Group 2. If you guys can all turn on your cameras, and your presentation is loaded up, you all may take it away.
Okay, hello everybody. So this is our Sequence to Success DNA barcoding presentation. We are RCC Group 2, and my name is Hilary Ortega.
Is Zen here?
Zen.
Sorry, I unmuted. My name is Zen Liu.
I'm Fiona Demaranen.
My name is Kimberly Vargas.
I'm Haley Salgonic.
And I'm Yasmin. Next slide.
Hi, so I will begin today by doing a little brief talk about the background of this experiment. So DNA barcoding was invented by biologist Paul Hebert, and this method is used to identify different species by sampling their DNA. For our experiment, we extracted DNA samples from various fishes, specifically focusing on the 16S ribosomal gene that is used to identify and determine species. We use this to determine whether or not the fishes were mislabeled or not. Next slide, please.
Some experimental methods that we went through: DNA extraction, PCR, which we're going to be going over in the next slide, which was used to amplify the target sequence. So we're making copies of what we're testing. We of course use the gel electrophoresis for a quality check, and we analyzed pretty much everything using DNA subway, which helped us compare all of our results. Next slide. So for our polymerase chain reaction, kind of broke it up into 4 different steps. Step 1, we're denaturing the protein at approximately 98 degrees, where the DNA strands melt and then they separate. In our stage 2, we're doing the annealing, which is approximately 52 degrees. This is where the primers are binded to the sequence of the target barcoding gene. After step 2, we go into step 3, which is extend. We're extending at 72 degrees, where the DNA polymers are now synthesized to a new DNA strand. And then from there, we enter step 4, which is we're repeating these cycles until we're ending with the final extension at approximately 72 degrees Celsius. Next slide.
So this slide goes over the information gathered from DNA Subway. As you can see on step 1, it shows the sequence viewing, which displays what our sample looks like. In step 2, you can see the blasting, which shows the most accurate species to go along with the DNA sequence.
Step 3 shows us the alignment of sequences that we compared with our DNA sample. The gray lines show us the sequence that is the same as our fish sequence, and the colored lines show where it's different. Next slide.
So after running our 14 samples within DNA Subway, our results concluded that 50% is correct, 21.43% is incorrect, and 28.57% is inconclusive. And you can see that each sample is followed below the proper category. Next slide, please.
As you can see, on the left is our group phylogenetic tree. It represents the evolutionary relationships between organisms. However, they're just hypotheses, it's not facts. Each branch shows the maximum likelihood of relationship of the species being tested to the most common ancestor. Next slide.
So to conclude, you're getting catfished. Our group in particular had 22% incorrectly labeled fish, meaning that 22% of our fish were mislabeled and as something different than it was advertised as. The reason for this mislabeling could be accidental, meaning it was just an honest misidentification, or the mislabeling could have been done purposefully, otherwise known as fish fraud. This can be done to cover up for selling illegal fish or fish that was sourced illegally. A restaurant might also do this with a financial motive as well by upselling cheaper fish and labeling it as something more expensive or sought out for. Next slide, please. The next steps would be to repeat the experiments we conducted. We had quite a bit of inconclusive data that was likely due to experimental error. So for example, during the DNA extraction, the proteins or the cell membrane might have not completely broken down to release enough DNA to be sequenced. So repeating the experiment will give us more accuracy and reduce any potential errors that might have occurred. Next, we could also increase the sample size since there was a lot of variation in the groups. We would combine all of our data so we could have more precise and cumulative results. We could also then focus on either specific species of fish or specific establishments like Joe's Sushi or Costco to see if there's any recurring patterns of mislabeling. Next slide, please.
As we know, DNA barcoding supports species identification, discovery of cryptic genetic diversity, and monitoring invasive species. DNA barcoding is very important for all these applications. Let me give you another example here, the identification of mosquitoes based on DNA barcoding. Mosquitoes dominate amongst other insects as carriers of pathogens that drive disease in humans, emphasizes how important correct DNA sequence identification is for mosquito surveillance programs and disease control programs. Researchers have shown the use of mitochondrial DNA, CO1, as a DNA barcode target and SNP analysis for mosquito species identification. This diagram shows us the procedure for the identification of mosquitoes using direct PCR. Thank you everyone for your attention, and we are happy to take any questions.
All right, thank you very much. And I will agree with Alex, very nicely done. Everything, yeah, transitions. You got everybody by telling them they were catfished. Very good. And yeah, it's kind of a standard question. What did you guys find most enjoyable about your experience?
I really enjoyed really applying the idea of the scientific method. So we learned about it all growing up and all in school and everything, and having the opportunity to actually participate in a real research project with our data being very valid, and even getting inconclusive results, having to do the next step, which is start from the beginning, get a new sample, go through the entire process again. It really gave me an idea of what a future and as a scientist, what we're going to be dealing with more research projects to come.
Yeah, I'll go with that.
I agree with Yasmin too. Also, I enjoyed, there's a lot of organization that we have to do with this experiment, you know, keeping all the data in the same area, analyzing everything. And I personally enjoy organizing things. So that was fun for me.
Also, I enjoy with my perfect, wonderful, awesome team.
I would agree with the rest of my group members. It was a terrific, it was wonderful experience, and it was nice to do this, although it was online, and to address Dr. Katie Burnett's question on what advices we have for future participants would be to take really good notes, especially in the beginning, because it gets a little bit confusing, and to have a good communication with the rest of your teammates, because at the end of the day, it's a group project and it takes all. So yeah, thank you guys.
To add on to that, I actually wanted to say also for like future participants that it's okay to ask for help, whether that's your group members or the leaders, you know, anyone. It's like this whole thing is to learn something new. So yeah.
Yeah, take advantage of your peer leader, because we were blowing up Nathan.
Yeah, we were.
Big thanks to Nathan. He helped us out a lot.
Yes.
Yeah.
Okay, thank you very much, ladies. And you guys got me with the Joe's all-you-can-eat sushi slide. That's probably one of the things I miss most during this entire pandemic. And I'm personally an Asahi fan, but I will take any all-you-can-eat sushi. And since lunch is coming up too. So again, very good job. Appreciate everything you guys did. And thanks for coming in looking all amazing. So if I can ask, you guys can all turn off your cameras now, and we will move on for the next group. Okay, so next we happen to have Moreno Valley College, group number 2, and you guys have all been upgraded to panelists. Feel free to turn on your cameras. All right, and your presentation is on the screen. The floor is yours.
Good morning, everyone. Good morning, everyone. My name is Sophia, and my teammates are Laura, Giselle, Yumena, and Ari. And we would like to present our 2020 DNA sequence examination. Next slide, please. So do you know how often fishes get mislabeling? Well, according to Ocean article, around 26 to 87% of fish are being mislabeled nationwide. 26 to 87% is frightening because often customers didn't pay for what they received, or worse, the incorrect fish leads to a serious allergy reaction. So one of our illustrations named that fish. As you can see, this species looks really similar, so it's really hard to distinguish which is which. So from that matter, we sequenced fish from multiple restaurants and markets to see if they were properly labeled. Also, you look at the New York data graph. My team got to examine salmon and cod. As you can see, salmon has 11 out of 45 inaccurate label, and cod has 4 out of 12. The percentage of correctly labeled seems really good. However, these fishes were still in the top 5 mislabeled in 2012. Therefore, in order for us to identify which fish is correctly labeled or mislabeled, we have to perform experimental methods by the following. Next slide, please.
Okay, so there are 4 total steps. So the first step, we need to get a fish either from a restaurant or a market, and we just get a small piece of a fish and we put it inside a tube, and then we just add the extraction buffer to the tube, and then we grind it really well and make sure there are no chunks. Then after the extraction, we just process the fish to the point of DNA extraction, which prepares it for the next step, which is to amplify DNA using PCR. So, which PCR stands for polymerase chain reaction. And in the tube, we will add where the fish DNA is at inside. We will add polymerase, nucleotides, buffer, red dye, forward and reverse primers. And the PCR will basically efficiently replicate the DNA at a very fast rate. That one DNA can give us billions of copies. Now on to the next step, the third step. Next slide, please. We will use the gel electrophoresis. So the purpose of this is just to get a visual representation of our DNA to ensure that we amplify the proper 16S ribosomal gene. And the reason we use this gene is because it is found in most living species and has enough diversity to identify a particular species. Now, finally, the last step, step 4, we use DNA subway. So since the whole project was done virtually, we use DNA subway in order to sequence and barcode the DNA, as you can see in the images below. So as a group, we input all of our fish, some top matches for each fish, and reference data into DNA subway in order to figure out our results, as you can see in the next slide.
After we barcoded the DNA, we found these results. As you can see, we have 4 fish that were properly labeled and 5 fish that were not. We had a sample size of 10 fish that were assigned to us, and out of those 10, one of the results came back inconclusive. This result was caused by a low quality score, which can normally be determined by the sequence having an error rate greater than 1%. This could have been caused by cross-contamination or an improper DNA extraction technique. Now looking on to the next chart. Considering one was an uncertain match, we disregarded that fish. We took the results of 9 fish that gave us 56% of the fish labels were not a match to their species, leaving only that 44% of the samples were correctly labeled, which is quite shocking because you begin to wonder what type of fish you have been eating this whole time. Now that we've discussed the overall percentages, let's take a look at the fish that were analyzed. We created an in-depth chart regarding the results. This detailed chart specifies the name of the fish, and we marked the correctly labeled fish. And as you can see, we also provided the correct fish species next to the samples that were incorrectly labeled. So the question that is left is, how much do the incorrectly labeled fish deviate from their actual species? We presented this using a phylogeny tree on the next slide.
So we input everyone's fish sample into DNA Subway, and we got the results via BLASTN, and we chose the top 2 to 3 fish species that most closely related to our sample, and we included it in our phylogeny tree, which you can see is a lot. So we narrowed it down to one of our fishes that were more closely related to its results. And here you can see that there are 2 branch points, and each branch point shows where there's a split in species. So to where it evolved into a new species, a new distinct one. And the more branch points between the samples, the more distantly related they are. So if we go into our next slide, we have our 3 fish species that were very far off from like what the results were. And you can tell because each of their results had 4 to 6 branch points between their samples. So with that, we can tell that they are very distantly related, and that's how we were able to tell that they were mislabeled from what they were advertised as. Next slide.
Okay, so all in all, based on our all examination of our fishes, we discovered about 56% were mislabeled and 44% were labeled correctly. Therefore, we can declare that fish that were given at restaurants and sold in markets are mislabeled. Also, examining all our fishes, cod, yellowtail, and pollock were deviated from their species significantly. And lastly, from our research, we also found one uncertain fish, Alaskan codfish, which turned out to be moonfish. This may occur because so their DNA sequence, it's may have contamination occur, which resulted in a sequence that could not be used. Also, in addition, we also did an extra research on the benefits of using PCR to identify DNA in the following slides.
Next slide, please. So throughout these 3 weeks, with the information we learned, we decided to apply it to the criminal justice system by looking at crime scenes such as homicides, John and Jane Doe, rape kits, and finding biological evidence such as blood, hair, skin cells, saliva, and seminal fluids, and then sequencing the DNA extracted. We can input the data into the National DNA Index System and the Combined DNA Index System. Next slide, please. So for example, we have the Golden Gate serial killer, Joseph DeAngelo. He was active in the 70s and 80s, but wasn't found until 2013 due to DNA evidence left at his crime scenes via GEDmatch. And for those who don't know, GEDmatch is an online service that allows the use of raw DNA to find related individuals. Next, to continue on with this, with genealogy websites, DNA is extracted through saliva using the same methods we used in the experiment. DNA is then sequenced and they look for genetic variations or SNPs, which are unique to every individual, like how fingerprints are unique to each person. This is how raw DNA can lead to finding an individual or their relatives. In the same way we found our fish species and organisms closely related to it. Any questions?
Okay, amazing job. Thank you very much. So, yeah, really great presentation, everybody. I really enjoyed your entire, the amount of work that you guys put in and the transition, as well as tying it down to potential DNA matches with the serial killer, Golden Gate Killer there. That was extremely interesting. Mr. Cortez would actually like to know, his usual standard question is, what did you all enjoy out of this particular project?
Not to be cheesy, but I enjoyed being with my group. They're very fun people, and I can say that I think we're friends now. But I also really enjoyed learning and like researching everything, especially about the Golden Gate serial killer. That was pretty interesting to me too.
Yeah, I also agree with Giselle. I really enjoyed like being with my group. Like they are fun people. Like the fact that I got to interact like in a group again, you know, because of COVID, we haven't really done that. So it was really fun. We have a really good group dynamic, and also how we were able to see the experiment in real life because all I've been doing is lobster. So it's all virtual. So it was really cool just seeing someone do it in front of me. But yeah.
Yeah, I guess for me it was like 4 of us go to MBC and Sophia goes to Mountain, another college. And yeah, and meeting her was pretty cool because even though I believe we all major in bio or biochem, we don't really know each other, and it was nice to meet everyone. Yeah.
For me, I'm actually from Crafton. I'm the only one from my school, like Laura said. So it's really nice to see someone from another school and just hop in with them. Also, I really enjoy putting data into the DNA subway. That's really my favorite part of all the research. It's really fun to see all the DNAs and how to see they give out like different name fishes, and then you have to do your own research and stuff. I really enjoyed that.
I agree with that. That was fun.
Okay, thank you very much, ladies. Again, amazing job. I think really great insight at the end in terms of also utilizing the technology of an online database for bioinformatics with GEDmatch. That's interesting. I guess everyone could now potentially input their DNA and you could maybe solve a crime and find a serial killer, or you could see if you were related to a serial killer. Either way, very interesting. Great job. And we'll see you guys at the end. So may I ask the this Moreno Valley group to turn off your cameras. And now we have our second to the last group. That's going to be RCC Norco Group 3. You guys are all added on. Please feel free to turn on your camera or your cameras. And I think I need to turn on one more for, let's see, I think Christian was there. Daniel, I will turn on your camera again. Okay, and then your presentation is up and running, so feel free to take it away.
Hi, nice to meet you guys. Good morning. All right, so we started our group. We're doing the DNA barcoding. It's my name's Daniel and Christiana, and then Paulina and Leah. We're all part of our group. And can we go to the next slide? So our background, the reason for this research is to see if we were actually getting the fish that was being advertised to us, because we all like to pay what we get with our money. So we want to get what we get paid for. No, we like to collect what we pay for. And then by doing this, we collected small samples of fish that were given to us for this research, and we processed them using the DNA subway. We used the 16S ribosome gene to confirm that we were getting the correct fish, the correct identity of the fish. And then the good way to figure this out is if we're actually eating what was being sold to us and advertised to us in the fish markets or anywhere we buy fish. The next slide, please. And then the problems that can lead to this is that are fish markets cutting corners? Are they doing this on purpose? Or are they just trying to save money? Or are they upselling on certain fish? Or are they just mislabeling them on purpose so they could just get rid of all the old fish that they didn't sell? And then there was a research conducted last year, and in Los Angeles, they took 119 samples and 55% were mislabeled. And tuna was the main fish that was mislabeled, which brings a lot of concern because that's more than half. And the next slide, please. And the question that we should all be wondering is that should consumers be worried about mislabeling fish that they are buying? Does mislabeling fish put people at risk? Can the FDA get involved? Is there any lawsuits to the market? Is there any huge health concerns? Is this a threat or not a threat to the consumers? And then can white flesh fish be more harmless than dark flesh fish? Is there specific safety protocols while handling specific fish, or is there no safety? Is there all the same handles, the same safety handles? And then after me, it'll be Christiana taking over the experimental.
Please, thank you. All right, so I'll be taking you through the experimental method for this group. And to start, I would like to go through the main steps for DNA barcoding. So first, we acquire a specimen, and in our case, a small piece of fish would be our specimen. And the next up, we're going to extract the DNA from the fish cells, and then we amplify the 16S rRNA gene using PCR, and then we check the results on an agarose gel. The PCR samples are then cleaned and sequenced, and finally the sequence is compared to an existing database. In our case, we use DNA subway to help with the comparison. Next slide, please. All right, so like I mentioned before, after acquiring the fish, we extract the DNA. Even though we didn't get to do this in person, we were able to watch our instructors do the experiment using the following methods. First, you label one of the 1.5 milliliters tube with the fish name and your number, and then you cut a piece of tissue about the size of your pinky nail and place in the corresponding tube. Add 750 microliters of extraction buffer to the tube and grind the tissue using the micropestle. And then you add 120 microliters of SDS to the tube after grinding. Cap the tube and mix. You place that into a 65°C bead block for at least 20 minutes. Then you're going to repeat the first 4 steps for the second fish sample. And then after that, you add 225 microliters, 5 molar potassium acetate to each tube. Mix well by inverting several times again. Make sure you mix well, and then you centrifuge the tubes for 5 minutes at top speed, approximately 14,000 RPM. Then you're gonna label each set of new tubes and then squirt about 700 microliters of the supernatant through miraculous funnel into the second tube. Next slide, please. Thank you. And then next, you're gonna add 600 microliters of isopropanol. Mix the content thoroughly by inverting and then centrifuge for 5 minutes at top speed. So the clue is to look for a small glassy pellet in the bottom of the tube that may be visible. And then carefully pour up the supernatant, then use a P20 set to 20 microliters to remove the large drops. Add 500 microliters of 70% ice cold ethanol and flick until the pellets comes off the bottom. Centrifuge again for 5 minutes at top speed, pour up the ethanol, then use a P20 set to 20 microliters to remove the large drops. Make sure the pellet stays in the tube. Place the tube in the bead bath with the top open for 5 minutes to allow the remaining ethanol to evaporate. And then lastly, you're gonna resuspend the DNA in 50 microliters resuspension buffer. Let us sit at room temperature for about 5 minutes. Then you can continue with the other steps, which is to check to amplify the RNA with PCR and then check your results with DNA subway. Thank you. And next, Paulina will be going through the results with us in the next slide.
So on this slide, this is a phylogenetic tree that we had with all of our fishes. Next slide, please. So 8 of the fishes that we got, 3 of them were mismatched. And 2 of them, the bakery fish was actually a channel fish. They're both catfish, but they're just different types. And then the next fish that we got that was mismatched was a swai fish. It was actually a rainbow trout. And although the rainbow trout has like red meat and the swai fish has white meat, sometimes the rainbow trout can have white meat, so that's probably why they can mismatch them. And then these results show us that we can't really trust our markets to give us what we actually ask for. And then I'm going to pass it on to Leah.
Thank you. After this presentation, we know that DNA barcoding is a technique used to identify organisms at the species level using DNA sequence. It is based on testing variations in natural DNA sequences, known as polymorphosis between these distinct species. Although there are many different uses of DNA barcoding, the one that stands out the most is the forensic application, since it is used to identify illegal animal trafficking in different parts of the world. Next slide, please. This application protects endangered species, preventing their hunting and sale in illegal markets. A research sample for this application is the article Forensic Application of DNA Barcoding for Identification of Illegally Traded African Pangolin Scales that is presented in the website titled Genome by the Canadian Science Publishing. In this research talks about how the pangolin species worldwide have experienced drastic population size reductions globally with the highest extinction risk in Asia, and also how DNA barcoding is used for law enforcement to be able to identify this specific species. In conclusion, it is interesting how we're able to analyze even one of the most easy but important things in our daily lives with DNA barcoding. Also, it is relevant to know that what we eat daily is labeled properly by the grocery store chains, and if not, incite them to be more careful with what is presented to the consumers. We'd like to thank everyone who made this opportunity possible: Dr. James Burnett, Dr. Katie Burnett, Mr. Alex Cortez, Dr. Chetty, Nate Tran, and our group leader, Isaiah Grant. Thank you.
Okay, thank you very much, team. You guys did an amazing job. Lots of great information. Christiana, that was a textbook tutorial. It sounded like you could work for a Food Network recipe, just going through the entire barcoding. I thought that was very enjoyable, and really great mention of the use of DNA barcoding for a penguin or the pangolin. Yeah, that's actually extremely concerning nowadays. I think it's from my home country of Vietnam. There's actually a big problem with that. So yeah, and it makes a lot of great sense to utilize this particular technology. So you will get the standard question from Mr. Cortez. What did you all find enjoyable about the project?
So I really like the tennis subway because it was so enjoyable. Like it was my first time using it, and I was so glad I got to do that. And I also like enjoyed the overall process of the whole thing. Like I learned a lot, and I'm really grateful for that.
For me, I really like the DNA subbank too, but what was most like interesting for me was watching the professors do the experiment on Zoom since we couldn't do it in person.
For me, that was a really interesting part as well, to be able to watch it. Although it's not the same as doing it in person, we were able to see the techniques, and also they were able to explain to us what were they using, why we're how was used, and all the things that were important. And also something that I really liked was to investigate and look for a different research which incorporates DNA barcoding to solve worldwide problems.
That this was I enjoyed it a lot. This was my very first research, and even though we couldn't be in person, I still enjoyed it. I really enjoyed the demonstration through the Zoom on our first day, and now by doing this really made me open my eyes and be more aware on what to buy, or what, like, if I'm really buying something, is it really what I'm getting? And but overall, I really enjoyed doing this research. Hopefully get to do more.
Okay, again, very great job. You guys did, yeah, you guys did amazing, well put together presentation. So you should all be proud of yourself. Okay, and then we will be moving on to our last but not least presenter. We have a solo act, Ms. Maria Sassati from Moreno Valley College. She was last woman standing for her particular group, but she was still able to manage to put together a presentation, and we welcome to listen to her. So Maria, feel free, turn on your camera. I'm gonna go ahead and leave the floor to you.
Thank you. Hello, everybody. I'm Maria Sassati. I'm from Moreno Valley College. I hope to transfer to UCR fall next year. As we all know that in the seafood market, products are mostly mislabeled, and we conducted an experiment using biotechnology, DNA extraction, polymerase chain reaction, gel electrophoresis, and I am going to walk you through the experiment and the findings from this experiment. Barcoding is the method of identifying fish DNA from a sequence gene in the DNA sequence. So the experiment, we are extracting DNA from a fish that we brought from the supermarket in order to compare the fish DNA to the DNA sequence in the database, that is the DNA subway. This comparison will allow us to decide if the supermarket is accurately labeling the fish that they're selling to us. We want to know if we are getting the fish that the supermarket is actually saying that they're selling. To go through the steps in the procedure, first we label the tubes to identify the specimen and add fish samples to the corresponding tubes, crush the specimen, and add 10% SDS solution in the sodium dodecyclyl sulfate with the deionized water, and place it in the bead block to protein in the DNA denatured at 65 degrees. Add 5 milliliters potassium acetate, invert the test tubes several times and place it in the ice for 5 minutes. Ice, centrifuge, and separate the supertenant. Re-ice the test tube for 5 minutes and the supernatant through the microcloth funnel into the second tube. Add 600 microliters of isopropanol and centrifuge after adding isopropanol and invert the test tube to thoroughly mix the contents and place them in centrifuge in top speed for 5 minutes. Remove the supernatant by carefully pouring it out and use micropipette set to 20 microliters to remove last drops. Add 500 microliters 70% ice cold ethanol and centrifuge after adding ice cold ethanol. Frequent chill the pellets comes off the bottom and place in the centrifuge in top speed for 5 minutes. Remove ethanol, use micropipette set to 20 microliters to remove the last drops. If placed in the bead block plate, place the test tube in the bead bag with top open for 5 minutes. This will allow remaining ethanol to evaporate. Resuspend DNA and 50 microliter suspension buffer and let it cool for 5 minutes in the room temperature. Determine assigned fishes. Each student are assigned two fishes. Each student are assigned fishes from the different market. DNA subway is used to analyze from different markets. DNA subway is used to analyze DNA sequence provided by the research members. Lastly, we analyze the results from using the DNA subway, and we will be able to know if the fish were actually labeled correctly. So to analyze from the data that we got from the experiment, first I want to walk you through the previous slide. I'm sorry, which is the DNA extraction. I want us to go through what we just went through the procedures. Thank you. So in case the procedures that we just went through, what we're trying to do is to extract the DNA. We were able to isolate the specific gene in the genome of the fish and sequencing this gene so that we can compare the sequence of the gene to known sequences of the fish. Whichever sequence matches our sequence will be the fish that we have. So all the steps that we just went through, we were able to isolate the DNA and compare with the DNA in the database. So for us to explain that the chemicals and the substances that we use in the experiment, we used, we first crashed mechanically to break down the cell wall, the cell membrane, the nuclear membrane through physical means. We use sodium dodecyclate sulfate to denature the organelle. Protein precipitation is achieved by protein acetate. Since DNA is insoluble, isopropanol and ethanol alcohol is added, followed by centrifugation, which forms a pellet of DNA protein. RNA are removed using chloroform. DNA precipitation is carried out using isopropanol and ethanol by dehydrating the DNA, and that's the precipitation. Next slide, please. So polymerase chain reaction is the automated version of DNA replication. It produces copies of a target DNA segment. In our case, it's the 16S gene. DNA polymerase is used to replicate target sequence, and chain reaction is because the DNA sequence is repeatedly replicated. The basic idea behind the PCR technique is that DNA denatures when exposed to heat. It doubles or expands. Once the strands are separated, we can copy each of them by primers, deoxynucleotide, and DNA polymerase. Primer is allowed to bind in the separated strand, which is known as annealing. DNA polymerase carries out the process of DNA synthesis, which is known as extension. As a result, we can get duplicated DNA sequences. This process is repeatedly carried out to make more copies of the DNA sequence. So we start from 2 strands of DNA, we get 4, 8, 6, and so forth. From this, the target DNA is amplified. Next slide, please. We use gel electrophoresis to separate DNA based on its size. Gel is made out of different substances such as agarose and polymerase. It is electro because we need an electric current flowing through the gel. From general chemistry, we know that in an electric field, current flows from the cathode to the anode where oxidation takes place. The buffer has different ions. We load our DNA on the well and it will flow through to the positive end. And whichever has the less weight goes the farthest. And the DNA ladder tells us exactly what side the fragments are. Example, we could have 400 base pairs and so forth. Next slide, please. From the data that we got from the lab, we found out that 20% of the fish was mislabeled. Mislabeling of the fish, it could undermine the consumer's trust, which has a negative effect on the seafood market. Improving traceability of the seafood will reduce mislabeling in the supply chain. Although DNA barcoding is expensive, it provides accurate information of the source of the fish. Next slide, please. The application of this DNA barcoding, we could use it to investigate in forensic investigation and fingerprinting. The DNA barcoding technique can be used to provide provenance information about a product. For example, by using this technique, we can ensure that products are not coming from endangered species like the elephants and the rhino. If we're buying ivory, we are able to know that is it legally harvested or where exactly is it coming from. This will help us to preserve the endangered species. Next slide, please. I would like to thank everybody who participated and also whoever made this possible. Mr. Nitran, Dr. Barrett Bodotham, Jim. I thank you all, Alex and Davante, my mentor. Thank you so much for helping me, especially that I did all this by myself. I just felt like it was a little pressure and you guys helped me out. Thank you so much, everybody.
All right, thank you very much, Maria. That will be a virtual round of applause for not only Maria, but all of you. I do want to commend you for really sticking through it. I mean, this is part of research and also part of teamwork. Unfortunately, you know, not your entire team was able to assist or come through at the end, but I appreciate your dedication to the project and program. All right, does anybody happen to have any questions for Maria? You get the obligatory question. Alex would like to know, what did you like most about this project and your experience?
What I liked most was the research, and I didn't know I could learn so much in such a short period of time. And also that the fact that I did it by myself, first I thought that it was a little pressure, and your willingness to help and the mentors being there and willing to jump in whenever they're needed. I think that was a very good example. And also it provided us with teamwork. Like I learned that you don't have to be a team for you to actually feel like people were there to support you. Thank you so much. Any other question?
Okay. I think that will conclude the end of our presentations. I would like to thank absolutely everybody here for their participation, for coming through and really sticking together as teams, as individuals. Katie is jumping on in. I'm actually promoting. So we could not have done this entire project without a lot of people helping out behind the scenes, as well as directly with you guys. So I'd like to take the opportunity to thank each and every single one of them. We had all the peer mentors, Gina, Isaiah, and Karina. We also had our peer leads, Devante with this nice LinkedIn profile picture, Nathan dreaming about Disneyland, Jesus, sorry, I didn't get a good picture of Jesus, so I tried to find the best looking one, and this looks pretty accurate, actually. The only difference is I think Jesus does look a little bit better. All right, and let's see. And then also, we have Ms. Katie Awashi from Moreno Valley College. She's the STEM Services Development Coordinator. She did a great job of recruiting and putting together a lot of this project and as well as our info session. So thank you very much, Katie, for jumping in and helping everybody out. Next, I think we have Ms. Patricia Gill. And Patricia, if you would like, you are welcome. Anybody who's been promoted to a panelist is welcome to turn on your camera and address the group or the students. But she's a wonderful STEM coordinator over at Norco College and has been a partner with us ever since the beginning. So many, many years ago. We've had so many great students from Norco College participate in a lot of the projects here at UCR. Very grateful to have that relationship and to have so many students come on through. And yeah, it's great to see you guys either on to bigger and better things, either at UCR or elsewhere. Okay. Next slide, we have my good friend, Mr. Dr. Scott Herrick. So I found his mugshot. Actually, no, it is a formal picture online. And I also have a picture here. This is the back of Dr. Herrick's head and me. And that's because Dr. Herrick's a huge supporter of this program, this project, as well as every student who comes, who participates. He normally shows up to just about all of our symposia. Today is no different. And this is just a picture commemorating one of the times where he was actively attending one of the symposia on campus for DNA Subway. So thank you very much, Scott. And of course we have Dr. D, or Darcy, as I call her. Dr. D, as you guys all know, she was a UCR alum and a huge proponent of giving this opportunity to community college students, as well as her students at El Camino. So I found her wonderful professional picture, but we had to scour the internet, not really our records, and I found a picture of her, again, supporting the students at one of the, supporting her students actually, El Camino, over at UCR for the DNA Subway Symposium. She's rocking the awesome CNAS shirt that I'm sure she loves so much. Again, thank you very much for your assistance. And then we have Dr. Meyer, amazing partner from Mount San Antonio College. I couldn't find any of her pictures, so I actually sat and made an avatar. Hopefully, and this is all from memory. Hopefully this is an accurate portrayal. So everybody from Mount Sac, you guys can attest to whether or not how that looks. But she's been an amazing program partner ever since last year when she actually came on, one of the first faculty to actually sit at UCR and go through this entire project. And now she's been a great mentor to the entire Mount Sac groups 1 and 2, and it shows in your presentation. Okay, Dr. Chetty. So she has her professional picture here. Thank you very much, Venki, for everything that you do and helping out with all the students, putting together the lab and answering their questions. Definitely could not have done this without her as well. So thank you very much. And we have Dr. Burnett, one of Dr. Burnett here. You're welcome to address the group if you'd like, Katie. Otherwise, yeah, we found her professional pictures and scoured the internet as well. That looks like an amazing super bloom out in the poppy field. But yeah, definitely could not have done this without Katie as well. She's been an amazing partner for many, many years. And she even used to fly down from Oregon and just to hang out with Mr. Burnett prior to their marriage and participate and help out every time they can.
Yeah, no, this is fantastic. Thank you very much, everybody. We couldn't have done this without you. We were really worried about how many students would we get. So I really appreciate that you took time out of your busy weekends to do this with us. So thank you. All
right. And of course, we have Dr. Jim Burnett. Here's his wonderful professional picture, and I was really happy to find in our trove of previous pictures, this was just last year, where we had all of you come on board, giving all your samples, and you could see Dr. Jim Burnett holding up 2 of the wonderful fish samples that you guys normally would have had to have analyzed and whatnot. Thank you very much, Jim. Do you have any words you'd like to say?
Yes, thanks. Great job, everyone. I really enjoyed listening to your presentations, seeing how you made it your own, even though they're all very similar. Sitting here with me is Sir Puck. I don't know if you can see him, but he says he likes fish. He's one of our 3 cats. And this is a very rare event for him to come and sit and let us pet him. But great job, everyone. Thank you to the community college partners who attended this morning and listened to the presentations, and to the STEM mentors and the students who led the projects. We do see this as a recruiting event, so yes, we hope that you see UCR as a potential to apply to and hopefully transfer to. If you have questions about that, Nee and his team are excellent at the application process, especially for the transfer students. So good job again, everyone. Hope you enjoy the rest of your afternoon, and back to Nee.
All right, thank you, Jim. And we have our last slide, last but not least, Mr. Alex Cortez. Would you like to say some words to everybody?
Well, first, thank you all for being here and dedicating 2 Saturdays to this, plus all the countless hours spent on the project. I think you learn a lot about yourselves in the process, and hopefully you're on your way as scientists and researchers. You got a taste of that. Hopefully it's not a fishy taste. And no, I did not eat all the samples, but that's just some fun. I wish I could have seen you all in person and kind of chance to interact that way. But I hope you stay in contact. I hope you do think about UCR as a potential school to transfer to, and go Highlanders.
All right, and then if we can pull out our Wizard of Oz behind the scenes, Ms. Gina Zio. She is hanging out at home. Thanks, Gina. Again, thank you very much. She's the one who really helped put together this entire symposium, in addition to our other peer mentors, Trina and Isaiah. But Gina is the one who downloaded all of your PowerPoints, curated them all, and has all this wonderful music in the back end. And she's the one obviously progressing all your slides. So she's probably going to have, a few dreams or nightmares, whichever way, for the next week, just thinking about next slide, please. So in any case, thank you again. Thank you, everybody. And just to show you our gratitude, I will be sending out an email just to ask you guys about experience. But more importantly, I want to get all of your current mailing addresses, because we will mail you a certificate of completion. So a formal certificate for your participation. And Dr. Burnett and I will be looking into trying to throw in a little bit of extra swag in there somehow. So we're not sure exactly what we'll be able to purchase under this limited time frame, but I will brave the trenches and go back onto our campus to mail all of these out personally.
We'll
so
send you all the
that
fish.
you guys, yeah, we'll send you some samples. We'll send you some unknown fish and some known fish, and no, that won't fare too well in the mail, especially during the holiday season. But again, thank you very much, everybody. It's been wonderful. It's been a pleasure seeing everybody, or virtually seeing everybody. We definitely hope to see you guys all personally on campus at some point. Dr. Shetty, thank you for turning on your camera. Would you like to say anything to the group?
This is first time I'm participated in this program, and really it's a great thing. Now whatever all the students, they have done their best. I enjoyed all the presentations. It was really good. Thank you, guys.
All right. Well, again, thank you, everybody. Again, just like Dr. Katie Burnett stated, extremely fantastic work, interesting and very wonderful and fulfilling to listen to everybody and work with everybody here.
And
Okay.
Neat,
Yes, Jim.
Yes. So we definitely need to say thank you to Neat. He does a lot of work for this project. You might not see him behind the scenes, but he does a lot of work and spends a lot of time, especially coordinating the STEM mentors and the mentors who helped you out. So let's thank Neat very much. And usually about this time, I hand him a 6-pack of his favorite beer, but maybe I can leave it somewhere in the mail room and you can pick it up when you go to campus. So.
Thank you very much.
Okay, I want to say something. Thanks, Dr. Burnett.
You're welcome. Yes.
Yeah,
Thank you.
it was really nice. Like, no, I enjoyed all the presentations. Like, no, it's great.
All right. Thank you.
Okay. Well, we ended at exactly 12 p.m. So works out perfectly. I believe it is lunchtime. After talk of all of this fish and food, I think this is a perfect time to end on a great note. So again, thank you to all. Great seeing you, and hopefully we'll see you back at UCR at some point in the future, but good luck.